To Srilata Organic Farming. This is a small scale Azolla production system. Earlier, we have seen the large scale production, that is in the ponds, in the water ponds in the field. But this is a very small scale system. This can be scaled up to the ponds. There is no issue. But while producing in the ponds, there is no this plastic sheet is not required. Directly in the pond, it can be produced. A water body is prepared by digging a pit of 2 meter length, 2 meter width, and 0.2 meters depth, preferably under the shade. This small scale unit, it will get heated up. That's why it is to be prepared under the shade. When it is going for the large scale unit without plastic sheet and all, that restriction is not there. Under shade restriction is not. Under shade restriction is not there. The pit is lined with a polythene sheet to avoid seepage losses of the water. This polythene sheet, either it can be single layered if it is thick one, or double layered. It is double layer or single layer can be used. About 10 to 15 kg of fertile soil is uniformly spread in the pit on the sheet actually. Slurry made up of 2 kg cow dung and 30 grams of not super phosphate here, because for the super phosphate there is no permission in the organic farming. The farmers who wanted to do the organic farming, they have to go for the rock phosphate. Mixed in 10 liters of water is poured in the pit. More water is poured into the pit to raise water level to about 10 centimeters. That much water is required for the azolla to grow. Not only cow dung, whatever permitted in the organic farming, any nutrition can be applied to it. There is no issue to apply any nutrition which is permitted in the organic farming. So, inoculate fresh disease free azolla, that is more important. Not only disease, pest free azolla is to be inoculated, because there are certain pests on the azolla also. 200 grams into the pond within 10 to 15 days. Azolla fill the pond and daily harvest by using sieves. Actually, daily some azolla, if it is harvested again, it will grow by next day. Daily like this, some azolla can be harvested. That can be composted to use to the soil, or that can be fed to the cattle also. Azolla is a good food to the cattle also. Replace the water and soil for every 3 to 4 weeks, and fresh nutrition can be added. If the growth is declining, again fresh nutrition, fresh soil and dung, these things can be added. Little cow urine also can be added, not more. But anything, any organic nutrition can be added to the pit, like sanjivak or anything can be added. Coming to the on-farm production of mycorrhiza. Mycorrhiza, we have discussed about producing mycorrhiza on beds. Here, let us see the production of mycorrhiza in pots. First of all, good mycorrhizal culture is very, very important. A starter culture, VAM. Whatever culture we are taking, the same will be produced. That's why good starter culture, which is having good infection capacity to the roots, and which forms a large root system, deep penetrating root system, bigger rootlets, that type of VAM is to be selected. And apply starter inoculum to the soil. The soil, before applying to the soil, if it is better to sterilize the soil, as mentioned earlier, in the hot sun or by heating the soil is to be sterilized. Apply starter inoculum into the soil, and then seed sowing. Seed sowing any seed, mostly maize or sorghum or millet. These things are preferred, so that a huge mycorrhizal growth can be obtained on these adventitious root system. Grow the crop normally how we grow the crop, the same way it is to be grown, like applying water and all. Chemical fertilizers and pesticides should not be given. Vermicompost can be given, FIM, these things can be given, but not the chemical fertilizers and pesticides. Once the crop is growing, uh, when it is at maturity stage, maturity means not full maturity, just when if it is maize, when tassels are emerging, that time, just a flowering stage, stop watering. So when watering is stopped, slowly it dries, the soil dries. During that time when the soil is dried, cut the top portion of the plant. That is not required. It can be used as the cattle feed or any other purpose. Only we want the roots. So after cutting, allow the pots for a few more days, like that only, without giving any water or anything. After that, harvest the roots. That means pull the roots, and the soil also is useful. Cut the roots and check whether the mycorrhizal infection is there or not. Under microscope also it can be checked, or visually also. Your experienced people will know by seeing just visually the mat like structures and all will be seen. Then the soil along with the roots, cut roots can be used, or roots alone can be used as the root culture, or root inoculum, or soil alone can be used as soil inoculum. But the best way is cutting the roots and mixing the roots and soil and using the entire thing is the good option. So this is about mycorrhizal production, on-farm production of the mycorrhiza. In the next session, let us see the biofertilizers, other biofertilizer production, carrier-based and liquid-based biofertilizer production. We will see. Thank you. Signing off, Srilata. Srilata Organic Farming. Let us see the biofertilizer production, like carrier-based and liquid-based biofertilizer production. Mostly in the market available ones are the carrier-based biofertilizers, which are very cheap and with less investment they can be produced. They are having very less shelf life. The disadvantage is temperature sensitive and contamination prone, low cell count, less effective. Liquid based biofertilizers are longer shelf life. They are easier to produce, temperature tolerant, high cell count, contamination free, more effective. Disadvantages: high cost. If you see the carrier based and liquid based, more than double the cost is liquid based biofertilizers are expensive. Higher investment for the production because fermenters and all are to be uh, arranged. So this is a carrier based biofertilizer. Mother culture is very important for any biofertilizer production. If mother culture is good, the end product will be good. If mother culture is having any contamination issues, then the end product also will have the double the contamination issues. So the mother culture is to be very pure, and then it is to be multiplied on shakers, initially on the shakers. Then the multiplied mother culture and shakers, it is to be added to the fermenters. Then fermenters, it will be ready, the biofertilizer will be ready as per the schedule, but there in the fermenter medium and all are to be added. There are various types of media for the production of the biofertilizers. The generally the carrier based biofertilizers are produced by companies which are having good infrastructure and all. So from the ferment, they are having their own media, or commercially available media are also present. Once the fertilizer is ready, then it is to be mixed with a carrier material like lignite or chalk powder, or so many carrier materials, talcum powder. So then it is to be tested for the quality. Quality means the count, purity, contamination. These issues will be checked.